Hello, my name is Reid Magnaghi, and today we'll be talking about how Dr. Oscar Tataroglu, a once postdoctoral researcher from the Department of Neurobiology at the University of Massachusetts Medical School, was placed under the Office of Research Integrity's scrutiny for his scientific misconduct. To better understand why Dr. Tataroglu performed the scientific misconducts he did, one must understand the background to his research. Previous research on the Drosophila flies showed that light was a direct indicator and effector for the timeless TIM pacemaker protein found in circadian rhythms for individual organisms. Circadian rhythms are physical, mental, and behavioral changes that follow a daily cycle. Environmental stimuli, such as light and temperature, have been proven to be primary causes for circadian rhythms. Being awake during the day and sleep at night is an example of light-related circadian rhythm. Circadian rhythms are important for they may lead to treatments for obesity, mental health disorders, sleep disorders, and other health problems. Previous research of circadian timekeeping has proven that the molecular mechanism used in Drosophila flies is similar to the one in humans. Dr. Tataroglu's research focuses on the temperature side and the SLL, which is small optic lobe dependent degradation interacting on the TIM protein, and how it affects circadian rhythm. Dr. Tataroglu's research methods included increasing the temperature from 20 degrees Celsius to 30 degrees Celsius. This elicits an intercellular calcium increase in circadian tissues. Down-regulating the TIM protein should result in a reset of the circadian clock in the Drosophila flies. This could be seen with temperature alterations. To better walk through this diagram, we can see that in response to thermal input, calcium and protease SOL trigger the TIM degradation. Thermal TIM degradation resets the Drosophila fly's circadian pacemaker. In mammals, the circadian thermal response can be due to the impact of the SOL homologous. The falsification of data grounds for scientific misconduct. Once research was concluded on any falsification data that Dr. Tataroglu had performed resulted in three major misconducts. The first one being the first paper of the Drosophila flies presentation saying their research concluded that temperature regulation and SOL degradation could result in altercation of the circadian rhythm in humans. He also falsified images and data on two other grants, along with data that was yet to be published. The data manipulations resulted in HP treatments overexpression of the protease SOL. Thus, Dr. Tataroglu selectively altered the original Drosophila behavior locomotor data in his primary data filings to support the hypothesis that temperature phase shift of the Drosophila circadian clock through regulated degradation of pacemaker protein, TIM protein, is mediated by SOL. Once Dr. Tataroglu's falsifications were found by corresponding author Patrick Emery and his team at the University of Massachusetts Medical School, Dr. Tataroglu entered into a voluntary settlement agreement. He voluntarily agreed to have his research supervised for a period of 3 years. This began on December 30th, 2019. Due to performing these scientific misconducts under different research groups and grants, he was no longer allowed to participate in any public health service supported research until the survey plan was submitted or approved by the ORI. Dr. Tataroglu agreed that he shall not participate in any public health service supported research until a supervision plan was submitted and approved by the ORI. If Dr. Tataroglu was to continue, a committee of 2 to 3 senior faculty members within his respective field would provide oversight and guidance. The sanctions put in place did not stop Tataroglu from being able to conduct any research. It just gave him plenty of extra steps in the middle to ensure that he was doing it ethically correct. Dr. Tataroglu was never barred, though his published work in 2015 was retracted in 2017. These falsifications impacted Dr. Tataroglu's career immensely. Once his data was proven falsified, he was fired from the university immediately. Through his voluntary settlement agreement, any institution that employed him had to go through extra steps to make sure that any information or data that he was collecting was precise and legitimate. Dr. Tataroglu was eventually brought on by the Einstein University in Istanbul, but he has yet to produce any further publications since his 2015 paper. More on top of the voluntary settlement agreement, Dr. Tataroglu could no longer hold any advisory roles, including board or peer reviewing, all included within the 3-year oversight period. Dr. Tataroglu's scientific misconduct does not only affect him, but also has repercussions for the scientific community. The money and time focused around Dr. Tataroglu's study could have been better utilized in non-falsified data. Instead, all these resources were pushed into Tataroglu's studies with no true factual base behind them. Before Dr. Tataroglu's 2015 research was pulled, it was cited in over 17 other related articles. All these cited articles now have far less credibility to them. On the bright side, the university and the corresponding authors do not look that bad due to them taking primary action and firing Dr. Tataroglu immediately once finding that he repeatedly misrepresented and altered primary data. And these are my references. Thank you.